Hey guys, my name is Didi Jimenez, and today I'm going to go over the chemical buffer systems that work against changes in the hydrogen ion concentration. Let's begin with the three lines of defense that occur due to changes in hydrogen. Maintaining normal alkalinity of extracellular fluids is highly important for keeping hydrogen balanced. In order for the pH of the body to remain within the narrow range of compatibility with life, generated free hydrogen must be largely removed from the solution while in the body. If alkalinity of extracellular fluid increases too much, there must be a mechanism to compensate in the situation. That brings us to our three lines of defense that work in order to maintain hydrogen stability of body fluids, despite any unregulated input. These mechanisms are the chemical buffer system, which is our topic for this presentation, the respiratory mechanism of pH control, and renal mechanism of pH control. Now, what exactly is a chemical buffer system? Well, it is a mixture of two chemical compounds in a solution with minimized pH changes when acid or a base is added or removed from the solution. The makeup of a buffer system consists of two substances that are involved in irreversible reactions. One of the substances is the yielder and the other is a binder to hydrogen. This occurs when hydrogen ion concentration begins to rise. An important example is a carbonic acid bicarbonate buffer. Which plays an important part in the reversible reaction shown above. That would be this example here. When an unbuffered solution receives a strong acid like hydrochloric acid, all this dissociated hydrogen remains within the solution. On the other hand, when the buffer pair H2CO to HCO3- is found in the solution that hydrochloric acid is combining with, the HCO3- rapidly binds with hydrogen to form H2CO3. The dissociation of weak H2CO2 occurs only slightly in comparison to the marked reduction in pH that happened when hydrogen remained unbound. In the case that the opposite happens, where the pH of the solution begins to rise due to addition of a base or loss of an acid, the yielding member of hydrogen of the buffer pair, aka H2CO3, releases hydrogen to minimize the rise in pH. Well, after that wonderful example, I now want to go over the body's four buffer systems, which are the H2CO3 buffer system, the protein buffer system, the hemoglobin buffer system, and the phosphate buffer system, which all serve an important role. Let's begin with the first buffer pair. Here I have all four of them. Just a little summarized version of it from the book. Okay, so the first buffer pair is H2CO3 to HCO3-, which is the most important when it comes to buffering pH changes caused by things other than fluctuation CO2- generated by H2CO3-. This buffer system is essential in extracellular fluid for two reasons. The first reason being that this buffer is abundant in extracellular fluid, meaning that it is available to resist changes in pH at any given point. The second and most important reason is that each part of this buffer is closely regulated. Within the body, HCO3- is regulated by the kidneys, while CO2 is regulated by the respiratory system, generating H2CO3. This means that this buffer system involves CO2 via the reaction CO2 plus H2O to H2CO3, which yields H plus HCO3-. With this equation here, there is a left side drive when hydrogen is added to the plasma from any source other than CO2. Once binding of hydrogen and HCO3- occurs, hydrogen no longer contributes to the acidity of body fluids, resulting in the rise of hydrogen to abate. On the other hand, if plasma falls below normal, for reasons other than a change in CO2, there is a drive to the right side of the equation. With all this being said, this buffer system works against the fall of hydrogen. Moving on to the protein buffer system, which is most important intracellularly. Protein buffers are the most plentiful in bodily fluids. This includes intracellular proteins and plasma proteins. These buffers are essential because they have both acidic and basic groups that can give or take up hydrogen. The protein buffer system, in terms of quantity, is the most important intracellular fluid because of the abundance of these proteins intracellularly. Most limited plasma proteins cause a reinforcement H2CO3- system in extracellular buffering. Moving on to our third one. Hydrogen that is metabolically produced from CO2 in transit between the tissue and the lungs is buffered by hemoglobin. The continuous diffusion of CO2 at the systemic capillary level occurs into the blood from the tissue cells where production occurs. With this large percentage of CO2 and H2O, we get the formation of H plus and HCO3-. Under the influence of carbonic anhydrase within the red blood cells, CO2 at the tissue level generates hydrogen, which binds to reduce hemoglobin and no longer plays a part in acidity of body fluids. Without hemoglobin, blood would be way too acidic, rendering it useless in picking up CO2 at the tissues. And now for our last one, consisting of acid phosphate salt, which donates hydrogen when it falls, and a basic phosphate salt accepts hydrogen when it rises. The phosphate buffer is most important in urine. This means that this buffer system can alternately change H plus for Na plus as demanded. And as seen by the book, it can also buffer ICF. After that quick summary of the body's 4 buffer systems, let's dig into the heart of the subject. When changes occur in the concentration of a hydrogen ion, guess what acts as the first line of defense? The answer, as you can already guess, is the chemical buffer system. In order to minimize the changes in pH, an immediate reaction from these chemical buffer systems happens within a fraction of a second. In order to compensate for the changes occurring to the concentration of a hydrogen ion, the buffer system's reversible chemical reaction shifts at once when hydrogen is altered. It is because of their fast response that this mechanism, aka our chemical buffers, are the first line of defense. From the time they are generated to the time they are eliminated, most hydrogen ions seem to disappear through the mechanism of buffering. Keep in mind that the chemical buffer systems themselves aren't eliminating hydrogen. The buffer systems combine hydrogen ions with one member of the buffer pair, which prevents them from contributing to blood fluidity acidity, and thus removes hydrogen ions. Because the limited capacity of these buffer systems to soak up hydrogen, there must be another form that the unseasonably produced hydrogen is removed. If an elimination of hydrogen did not eventually happen, all of the buffers penetrating to body fluid would be bound to hydrogen, leaving no further buffer activity. The respiratory and renal mechanisms are the other response mechanisms controlling pH and getting rid of bodily acid instead of suppressing it, resulting in a much slower response time in comparison to chemical buffer systems, but those are topics for another time. Let's move on to the clinical significance. Okay, so its role in physiological processes makes acid-base balance in the human body an important paramount. During respiratory acidosis, cellular respiration produces CO2, which projects into the environment. In the body, the formation of carbonic acid occurs when carbon dioxide and water combine within the body via carbonic anhydrase. This carbonic acid dissociated into a hydrogen ion and a bicarbonate. With there will be a decrease in pH due to a reduced respiratory rate, meaning more CO2 exhaled equals less CO2 available for this reaction. While this isn't the only disorder, this is the one I chose because it occurs because of hypoventilation and can be due to multiple causes like severe obesity, brain injury, and chronic obstructive pulmonary disease. Once it occurs, the correct metabolic response is to increase the amount of bicarbonate through the renal system, which unfortunately doesn't always happen. I also didn't find a case study on it, so I decided to just summarize it up. So that's that. In the resources, you'll find the link to the article about this, which was very interesting. It mentioned other examples of the clinical significance. Well, that's a wrap to the presentation, and those are the resources. The top one is the article that I used, if you want to look further into it. Thank you guys, and I look forward to seeing you on the next presentation.